Hi, my name is Ashley Jacobi, and I am a scientist in the Research and Development Group at Integrated Technologies. Today, I'm excited to present some of the new work our team has done in the CRISPR space, specifically focusing on some reagents that we've developed to enhance CRISPR efficiency and specificity, and then to showcase a new CRISPR analysis pipeline that we've developed that we hope to release out to the community soon. So in our R&D group, we've really focused over the last 5 years to develop innovative solutions for the full CRISPR workflow, starting with the design of our reagents all the way through to analysis. And today I'm really going to focus on the things I have highlighted in orange, which are some of the newer things that we've developed over the last year. But to kind of highlight everything, we have an incredible bioinformatics team that works very closely with our wet bench scientists. So we have taken empirical data and developed an CRISPR-Cas9 guide RNA design tool. We also, earlier this year, launched an HDR design tool, which has a nice user interface and integrates with our guide RNA design tool, providing rule sets to develop, help you develop your HDR substrate. And then we also have our RAMP-seq design tool, which will help you design primers for NGS analysis. And then in our reagents category, we've done a lot of work focusing on guide RNA development and protein development, and I'll highlight some of those today as well. For our HDR reagents, we've got short modified single-stranded oligos, as well as longer fragments, and we're working on a new technology where we're modifying double-stranded fragments to get you a little bit longer material. And we've been doing a lot of work with small molecules to study those and see what of those will help boost HDR rates. And lastly, we've really put a lot of effort into optimizing our RAMP-seq system for CRISPR. And this is an NGS multiplexed amplicon sequencing technology that I'll spend a bit of time talking and linking that now to an internally developed NGS analysis tool that we are hoping to provide through a cloud-hosted environment next year. So jumping into some new developments with guide RNAs, our team has spent a lot of time optimizing the length and the modification patterns for both a two-part and a single guide RNA for Cas9, as well as the cRNA for Cas12a. We've done a lot of this work in R&D, studying the length and modification patterns, but we've also done a lot of optimization in our manufacturing group to make sure we are able to synthesize the highest quality material and keep pushing the lengths that we're able to make. And since we are able to make these synthetic guide RNAs in high throughput, we are able to provide a lot of customizability to researchers doing genomic screens, knockout loss of function assays. And so we can provide CRISPR libraries by tens of thousands of guide RNAs. And we do have some stocked options where we've got whole genome library, human kinase library, and a human epigenetics library ready to order as our two-part system. But we also do a lot of custom library synthesis for our customers that can be done in two-part or single guide RNA as pooled or arrayed format. And this takes into account utilize our IDT-derived design algorithm that I mentioned has come off empirically defined data to help predict the best quality guide RNAs, or we can take custom designs into effect too. And so highlighting some data we've generated internally for CRISPR screens using different guide RNAs. In this case, this is just a small experiment here shown where we've looked at 4 guide RNAs in 5 different genes synthesized as either a two-part guide RNA for Cas9 or a single guide RNA. And what this figure is showing in the top panel is the two-part guide RNAs, and the bottom is the single guide RNAs. And we've delivered these via electroporation into HAP1 cells at 2 different doses as RMP complexes. And so the green is going to be the higher dose, and the blue is the lower dose, where the line is the average, what we're seeing. And what we really see here is that at the higher dose at all of these genes, for both the two-part or the single guide RNA, we're getting greater than 85% efficiency at every loci we're looking at. And this is in HAP1 cells here. So, you know, our algorithm is predicting very high efficient guide RNAs, and we're able to deliver these and create really high levels of editing. Another thing I wanted to just touch on, as the CRISPR system is expanding and new nuclease systems are being discovered, we wanted to make sure we make it easier for researchers to be able to get RNAs of any length. So soon we'll have a custom guide RNA ordering tool that will allow you to order guide RNAs from 30 to 150 nucleotides right off our website, supporting chemical modifications. And this will allow you to study these novel systems and be able to have a really easy way to order these and get you guide RNAs that are very customizable to your experiment. And so likewise, in our R&D group, the great new technology that was published last year out of the Lu Lab, prime editing, we wanted to study this using chemically synthesized pegRNAs at IDT. And so prime editing functions through genome editing without a double-stranded break, nor the need for a separate donor DNA template. So what the prime editing construct is a fusion protein of Cas9, H840A nickase, and an MMLV reverse transcriptase that then associates with a long guide RNA known as a pegRNA. So that is shown, the pegRNA is shown here in the blue, green, and orange schematic. And so this ends up being in the range of 120 and greater length RNA, which is composed of the targeting RNA region, which is that lower dark blue region, and then the enzyme binding region in green, and then a region that has the primer binding site and a region where you want to make your corrective material. So what we've done here is we wanted to study how chemical modifications in the pegRNA would tolerate to prime editing. So what we have done here is we are inserting that CTT sequence at site HEC3, and we are using either unmodified pegRNA or a chemically modified pegRNA. And the sequence for this is shown on the bottom, where we've put three 2'-methyl methyl bases and 3 phosphorothioate linkages on each end of this 122 nucleotide RNA oligo. We've electroporated this into K562 cells along with the PE2 plasmid, co-delivered this. And what you can see here in orange is that there's no insertion of that CTT assessed via NGS when we've used an unmodified pegRNA. However, when we use a pegRNA that is chemically modified in the blue color, we are seeing high levels of insertion comparative to just delivering this all as a plasmid construct. And as a proof of concept, we have also purified this PE2 construct as recombinant protein, and we have complexed this to the modified pegRNA and delivered this with or without our ALTAR Cas9 electroporation enhancer into GERCAT cells. And we do see successful prime editing in a dose response here by using delivering this as a ribonuclear protein complex. So to kind of now focus a little more on the CRISPR protein engineering updates that we've done, our R&D team really has focused over the last couple of years on 2 main things in our protein engineering R&D group. The first point is to improve fundamental shortcomings of the popular CRISPR enzymes, the main one being Cas9 from strep pyogenes. And the main identified problem with that is that the editing is not perfect and we can have some off-target editing. So we have, through bacterial mutagenesis, evolved a high-fidelity Cas9. And then AsCas12a or LbCas12a, the main shortcoming of that protein was just the lower on-target editing seen when this is delivered as a RNP complex. So we also did a bacterial mutagenesis screen to evolve an enhanced activity version. Some other things we've been working on lately is to develop new CRISPR enzyme formats to basically broaden the utility. So creating some fluorescent fusion proteins where GFP or mCherry are tagged to the protein, and then creating some enzymes that are in alternative buffers. So specifically a glycerol-free storage buffer, which functions as well as our standard 50% glycerol storage, but is amenable to liquid handling. And in some cases, we've seen higher performance. And then it could be a single component storage reagent as well. So to highlight some data from each of those proteins that we've developed, a couple years ago, the big focus was on developing the high fidelity Cas9. And this was published in Nature Medicine. And briefly, what was done here was through low fidelity PCR, creating a bacterial mutagenesis library where we set up a two plasmid system where the mutants we were screening had to cleave the on-target site and not cleave an off-target site to survive. So doing that screen, taking the mutants that survived that and purifying those, and then actually studying those in the context of an RNP delivery and seeing which one ultimately maintained on-target activity while reducing off-target activity. And what we've settled on is what we refer to as the ALTAR Hi-Fi Cas9. And just a one-figure summary of this is shown here on the right, where, in a worst-case scenario where you're working in an expressed system, what this figure on the left is depicting is, in orange, the total editing we're seeing at the on-target site. And then every blue bar is editing we're seeing at off-target sites. So if you take all that total reads together, from that, the population actually has only 28% of on-target editing, where the remainder of what we're detecting is all different levels of off-target. So then switching and using our high fidelity Cas9, we're able to maintain high levels of on-target activity and almost reduce all of that off-target editing. And this is at a guide that is has very high levels of off-target editing. And further, what's shown in that manuscript is that this high fidelity Cas9 is able to provide robust sickle cell gene correction. So in that top panel A, we're able to show maintained activity of the Hi-Fi Cas9 when delivering alongside an AAV6 construct. And then in panel B, the on-target or the off-target levels, you can see there that off-target 1 has 30% level of editing with the wild type, and we're able to reduce that with the Hi-Fi Cas9. And additionally, I wanted to point out that the Hi-Fi Cas9 is available as a GMP protein off the shelf through Aldebaran, trademarked as SpyFi Cas9. And so moving on to the other directed evolution project that we have done is for AsCas12a in a similar manner, where in this case, we are wanting to identify a construct that had higher activity. So we set up this system to find a construct that could cleave a quadruple T, which is the PAM site that is least active in Cas12a. And what we've settled on is our AsCas12a Ultra mutant that now has superior editing levels over the wild type Cas9. And that's depicted here in this figure where we've looked at 71 different sites. And what's shown in this box and whisker plot on the left, in HEC293 cells, we're comparing the wild type Cas12a protein complex to the guide RNA and delivered as RNP. Those 71 sites, you can see the average editing of all of those sites is under 40%. However, now with our Ultra, we're near 100% editing. And additionally, as I mentioned, we work very closely with our bioinformatics team. So now we are, going even further now to try and design an algorithm to isolate out any of those low performing sites. But you can see by just random selection of guide RNAs, we are getting very high levels of editing now with this Ultra. And then the second side of this graph is showing in Jurkat cells, comparing this now to Cas9 and looking at guide RNAs in similar genomic regions, where we're able to now see editing levels that are very comparable to Cas9 using Cas12a Ultra. And a serendipitous finding we found with our Ultra mutants for both AS and LB is that they actually have increased temperature tolerance. So here we're delivering RNP complexes into cells and then incubating them at either 30 degrees Celsius or 37 degrees Celsius. And in a dose response, you can see that at 30 degrees, the Ultra variants have much higher activity than the wild type and are much more tolerable to these lower temperatures. As I mentioned, we've also recently developed a glycerol-free storage buffer that maintains activity of both our Cas9 and Cas12a suite of proteins. And so here we're showing some data where we have delivered about a dozen different guide RNAs into HEC293 cells via electroporation, where the protein is in our standard 50% glycerol buffer or now in a glycerol-free buffer. And we've delivered these at 2 different doses. So on the left, we're showing the individual data points or individual guide RNA sites in blue and orange is at the high dose, and the other 2 colors are at a low dose. And what we're seeing here, which is shown probably a little more clearly in the right figure in the box and whisker, is that the glycerol-free at both doses has equal activity, if not a little higher. So we're able to maintain that activity now, storing this in a glycerol-free buffer. And then the last protein that I'd like to highlight today is working on developing fusion proteins where Cas9 is fused to GFP or an M-cherry to allow for visualization of your delivery. So here in the bottom panel, we're able to see, we're able to image the GFP in our HEC293 cells where we've delivered this RNP complex. And now doing some facts and looking at enrichment of editing from using this. We've done this for both nucleofection and lipoffection. And what we've done here is we are looking at the editing levels of the unsorted population of cells or population of cells where we've taken the top 20% of the GFP expressing population. And what you can see in the bar graph is that, especially for lipoffection, we are seeing a nice boost in total editing when measuring the editing levels of the top 20% that we've been able to sort out from that GFP protein. Nucleofection, you can see a slight boost as well, but we've seen that nucleofection is so efficient already, and you can kind of see that in the lipoffection having a broader distribution in that FACS plot. But using this does really enrich the editing levels, especially when you are delivering via lipoffection. And then to show that we aren't seeing any loss in activity from these fluorescent proteins, this bar graph shows here in the top panel for Cas9, the blue bar being wild type, so your baseline, delivering these as RNP into HEC293 cells. And then the other 2 bars showing the level of editing when the Cas9 is fused to GFP or M-cherry, you can see is very similar levels of editing. And the same sort of thing on the bottom panel where we've done this for Cas12a, comparing our Cas12a Ultra unlabeled protein to now our fluorescently labeled GFP protein, very similar levels of total editing. And since we're discussing fluorescent reagents here, I did want to mention our We also, if you don't want to label the protein, we can also label the tracer RNA if you're working in a two-part system. And so we've got these reagents where we've found putting a 5' fluorophore on the tracer RNA. So that's shown in the schematic off to the right. You can see that kind of red firecracker looking image is where the fluorophore goes. And so we've done this with 3 different dyes here. We've got an ATO 488, an ATO 550, and an ATO 647. And you can see in the FACS plots how we're able to isolate out those populations that have that fluorescent tag on the tracer RNA. And then doing a similar sort of thing that I just showed with the protein, we are now able to, when delivered with liposuction, we are here comparing at 2 different guide RNA sites, the unsorted population in that set of 4 bars. The 4th bar on the right of the set of 4 is unsorted. And then if we take the lower percentage of the particular wavelength we're looking at, the medium percentage, so the 60 to 80%, or the top 20%, we're able to show enrichment of editing for all. Again, we've shown here the GFP protein again is the first set of 4, and then the next 3 sets are looking at the 3 different tracer RNAs. So you can see for one site on the left, that 231 site, and another genomic loci on the right, that we're able to get enriched levels of editing using these fluorescent, either protein or tracer RNA, when we've delivered the RMP complex, in this case with RNA IMAX. Next, I wanted to highlight some of the improved solutions that we've done for HDR. The last couple of years have been working on studying different small molecule compounds to add into your tissue culture media after delivering your electroporating your components into your cells, and then incubating that in media that contains these HDR enhancers. So we have what we refer to as our HDR enhancer V1, which if you compare that to the gray bar, which is a control, here we're looking at HDR rates where we've made a small change, a 6-base insertion, and then measure that perfect HDR by NGS. And so the gray bar shows the HDR levels we get from just a control. And then if we add in that HDR enhancer V1 in blue, you can see the boosts we're getting in 4 different cell types for both Cas9 and Cas12a. And we've now recently discovered another HDR enhancer that we're able to add at much lower doses. So we're adding this at 1 micromolar compared to 30 micromolar, and see even further boosts of the HDR rates. One other piece of work that we've really focused on is defining a rule set to generate the highest rates of HDR and building this into our HDR design tool. And this work here was done by Molly Schubert in our lab and is out for review right now, but where we've developed an empirically defined rule set for where to place silent mutations. So if you're adding an HDR event in a place that doesn't disrupt the protospacer, we have found that adding in silent mutations can really boost the levels of HDR. And so to show as an example here in the top kind of grid figure that shows the different red and blue boxes, what we've looked at many different sizes of insertions and deletions. And if we do not put a silent mutation in, the total level of HDR in this example at the very top, that SAA1, is coming out at 0.6% of that insertion of that T right there. And then this is showed through conditional formatting, but where we make the silent mutation, the blue coloring is where we're getting higher rates of HDR. So we've done this for, as I said, for many different types of HDR events that we want to create, and have developed a rule set that we have integrated into our HDR design tool. And a nice summary of that is in the bottom right corner, where we're looking at different types of HDR events without silent mutations and the boost you get with adding silent mutations. And so as I mentioned, that's all included in this HDR design tool, which it was a lot of work between our bioinformatics team and our wet bench scientists to develop this, but it's a novel bioinformatics tool for single-stranded HDR template design that also interfaces with our guide RNA selection tool. So you're able to indicate where you want to make a correction, and this will add in silent mutations if that's what you choose. This is supported with wild type Cas9 as well as a nickase, and will then can be ordered with our ALTAR modified donor oligos that also are modified to further boost activity. And so we've spent a lot of time really focusing over the last couple of years of developing rules and reagents to increase the rates of HDR for shorter knock-ins, but now we are also spending putting a lot of effort into how can we further improve large knock-ins. And so one thing we're focusing on right now is coming up with solutions to reduce blunt integration if we want to use double-stranded donor templates. And so this blunt integration, after you make a double-stranded template, there is risk of, through the non-homologous end joining pathway, those homology arms essentially being put into your double-stranded break. And to avoid that, what we've been studying is using modified double-stranded donor templates, similar to work that has been published, expanding the modifications that we're looking at. And what we're showing here is that looking at 300 base pair insertions, a 500 base pair insertion, or 1,000 base pair insertion, and comparing an unmodified double-stranded donor template to our ALTAR 1 modified template or ALTAR 4, two different types of modifications that we're studying right now. And we're measuring HDR insertion rates via MinION in this case. And we've got 100 base pair homology arms on each side. We've delivered these as RMP complexes, and the figure here is showing in the solid bar portion the HDR rates, and in the striped portion the blunt insertion that we're seeing from those unmodified templates. And so you can see at all insertion sizes, we have reduced that blunt integration rate by using these modified donor templates. And we are continuing to study this, and this is something we're really excited about. And coupling together this new HDR enhancer I just talked about with these modified donor templates, we show here the additive effect you can get of these two improvements, where in blue is a modified donor template for a, again, 300, 500, or 1,000 base pair insert, compared to in blue with no HDR enhancer added, whereas in gray, we've added the HDR enhancer. So we've used a modified double-stranded donor template with our HDR enhancer, our new version, and in this case, we are seeing up to 60% HDR rates for these large insertions. So the last thing I want to focus on here is our analysis tool that we've developed, and specifically focus on how we analyze our CRISPR editing events through using our RAMP-seq technology. So as I mentioned, and has been well published, gene editing is not perfect. CRISPR can result in edits at unintended places in the genome. And so we need a way to measure these on-target as well as these off-target events, be able to identify and quantify them. So I want to talk through the workflow that we use in our lab for detecting and quantifying off-target editing events. So an example workflow that we go through is first to nominate the off-target sites. And this can be done through different methodologies, through GuideSeq, through an in silico prediction tool, through other in vitro assays. But once you get a nominated list of sites that are predicted or have been empirically defined through a GuideSeq type technology, we then make a a panel using our RAMP-seq technology to then do amplicon sequencing and quantify in your cell type in high throughput the actual level of editing. And then we have a data analysis pipeline that we use to accurately quantify those levels of editing. So to break that down a little more, the first thing that we need to do is identify the off-target sites for a particular guide that we're looking at. And so, as I mentioned, there are unbiased detection methods that you can use. There are many different in silico prediction methods. The commonly used guide-seq technology out of the 2015 publication, you'll get a readout like this, which using your guide RNA of interest will show you other areas where that double-stranded tag has been integrated. So we take a combination of these unbiased detection methods with in silico prediction methods, and we come up with a list that we then use the RAMP-seq system to quantify these off-target sites. So a little bit of background about what RAMP-seq is. RhPCR is something we've studied at IDT for many, many years, even decades. And what this is it is a PCR primer that contains an RNA base, which is shown in blue here, and contains a blocking group. And so the way this works here is then our PCR contains Taq polymerase, as any amplicon-based PCR would, but it also has RNase H2. And what RNase H2 does is it recognizes this RNA base and cleaves 5' of it, leaving a hydroxyl group for amplification to then a 3' OH for amplification to then take place. And this is able to then create very specific PCR. And so we're able to leverage this and do very specific PCR, do multiplex PCR. And so to highlight the technology greatest is to reference you to the 2011 publication that came out of our group. But the schematic here shows nicely how this PCR works, where we've got our primers that will anneal to your target, and they've got the RNA base and a blocking group. And that RNase H2 recognizes that DNA-RNA hybrid and will then cleave and hybridize the primers, allowing DNA polymerase to extend the unblocked primers. So as I mentioned, what this allows you to do is have really great specificity and a much lower potential for primer dimers. And so that's depicted here in this graph, or in this gel image on the right, where you can see when using 10 nanograms of genomic DNA here, the amount of primer dimers you're seeing in that bottom band with standard primers on the left, or when using the two lanes on the right, how you're not detecting those primer dimers when you're using the RhPCR primers. So what this allows you to do is to have much higher multiplexing capabilities, where we're able to put up to 5,000 assays into one panel and do one reaction. So it's really amenable to studying a lot of different target loci at once. So we've specifically integrated this RAMP-seq technology into an NGS solution that we are now using for CRISPR editing. And so what this all comes with is an optimized primer design tool. So we've our bioinformatics team has worked to make a nice user interface where you can order and have your PCR primers designed with this RhPCR technology by simply entering your bed coordinates, and then you get your primer designs for your on-target site for up to 5,000 off-target sites. We've developed a high throughput library prep workflow, which is depicted below, which you have your edited genomic DNA and then your pool of primers, and that red circle in those pools kind of indicates that these are blocked primers. And so you do 2 rounds of PCR for this amplicon sequencing. And so the first round is locus-specific PCR, where the RNase H2 is present. So this will allow you to target many regions at once and not have the risk of the primer dimers. PCR2 is then doing your universal amplification, adding on those adapters, and then these are sequence ready to go for Illumina sequencing. And then we have taken our internally developed analysis pipeline, and we are working now with a third party to make this available through a cloud-hosted environment. So as I mentioned, our RAMP-seq, we have a RAMP-seq design tool that will support many different reference genomes. You come with a .bed file, and then you can input this in, and you'll get designs for your up to 5,000 assays. We'll let you know how many of these can be pooled together and in what size panels. And then showing some sequencing data here. By using this technology, we're able to get very high quality sequencing data, and looking at metrics here as far as percent mapped reads, on target rates, and coverage uniformity for different panel sizes when we're using 10 or 50 nanograms of, in this case, Coriell genomic DNA samples. The set of bars on the far left of each graph is a 19-plex assay panel. So we've got 19 different assays in that pool doing just one PCR reaction. The second plot or second set of bars is for a 282-plex and then a 994-plex using our high throughput protocol or our regular protocol. And the high throughput allows you to only have to do one spray cleanup, but you can see here, great levels of uniformity and on target rate. So this is really, really great and allows you to do your library prep in high throughput. And so just to kind of highlight back to that figure that I showed when, where we were developing the hi-fi protein. So the figure that went into that publication used the RAMP-seq technology. So in this case, we are using this technology to validate our high fidelity Cas9. And so that panel on the left where we see many off-target sites, those were all nominated through GuideSeq, and then we came back and quantified this with RAMP-seq. So in this case, we're looking at, in all of the different conditions we tried, we were looking at essentially over 1,500 different individual PCR reactions that we were able to greatly reduce by running these panels of assays together. And so this is just really great for reducing reagents, reducing time, and able to produce great validation data for this high fidelity protein. And the RAMP-seq technology now has been published in many different journals now, and this highlights some of the different publications where people have used this technology to assess their off-target editing. RAMP-seq can also be really useful for genomic screens or large knockout studies that you're doing, because the RAMP-seq design tool can do all of the PCR design for you. You then do your delivery, your library prep, and you can do your data analysis. And in this case, we used that for when we were validating the Cas12a ultra protein. So we're looking at 120 different sites in Jurkat cells and 96 different sites in 293 cells, and trying to see at these different PAM sites how much better ultra has gotten. And we have done this all through our RAMP-seq NGS analysis, design, library prep, and our internal analysis tool. And so to mention that analysis tool and kind of bring us back to the workflow. So again, the first piece is to identify your off-target sites, and then to do your quantification through RAMP-seq, NGS amplicon sequencing. And then now to analyze that data, we've built a tool that we refer to as CRISP alterations, and are partnering with a third party to build this user interface where you're able to simply do a drag and drop of your FASTQ files into this and get your target report readouts, your analysis through visualizations. And to highlight how all the back end of that is working and what we're doing here is the pipeline is doing the sequencing QC, it's doing the adapter trimming, the quality filtering, the read merging, we're doing the filtering and the binning, the target alignment, and then what comes out here is then, you can kind of see some representations of this on the right, the visualizations of your endo profiles. This is compatible with IGV. This will show you perfect HDR. So this has been incredibly useful, and this will, we are working to get this put out in the cloud and be provided for you with RampSeq. And so to show some functional data of our internal pipeline, what we've done here is we are using a synthetic data set where we know the editing is 50% of a lot of different target types. We've looked at 603 different target spaces right here, and we are comparing what our tool predicts as the editing, knowing the truth is 50%. So in green on the far right of the graph shown here on the left side of the slide is for Cas9. If we focus on that, is the CRISP alterations tool compared to some other tools that we've tried out there, and just the high accuracy we're getting from this analysis pipeline for both Cas9 as well as Cas12a, which is shown on the right-hand side of the slide. So with that, I really want to thank the fantastic functional genomics team at IDT that has done all of this great innovation work to really try to improve every step of the way of the CRISPR workflow and keep coming up with innovative technologies for this. So with that, thank you for your attention, and I'm happy to take any questions.
All righty, awesome job. Well, we're all set. So pretty much from here, I'll just do some quick editing on the front and back ends. And then since there's no interruptions during your presentation, we don't need to do any editing there. And then I will have it ready for the event next week. And I'll also send a preview link once the editing's done, so you can take a look at that.
Okay, sounds great.
Awesome. Any questions for me before we wrap up?
I don't think so.
Okay, sounds good. Well, if anything comes up, feel free to reach out. If not, I will send that link over once it's ready.
Okay, sounds good.
All righty. Have a great weekend. Bye-bye.
Bye.